For many people, scientific testing with animals is something that makes them uncomfortable. About 140,000 adult fish are used in Europe annually to test chemicals for toxicity. Tests are required for all chemicals produced in volumes above 10 tons per year before they go to market. Fish are also used to test the toxicity of effluents and wastewater. An estimated 3 million are used in North America each year for these tests. Is there a good replacement for testing with adult fish? Are there good alternatives? Scientists at Eawag contribute to finding out whether there are other ways to do these tests. One such method is to replace adult fish with zebrafish embryos. In fact, there is now such strong scientific support for the use of embryos that the OECD is adopting fish embryo testing as an official test guideline in the autumn of 2013. Embryo tests can be used instead of adult fish acute toxicity testing. Melanie Knöbel is one of the research assistants who worked on determining the effectiveness of embryo testing.
I investigated how embryos can best replace the use of adult fish to test chemicals for their acute toxicity.
Melanie and her colleagues carry out these tests in the labs at Eawag, where scientists keep adult zebrafish in fish tanks to get eggs from the fish. The trays are covered with a mesh and green artificial plants are attached and marbles placed on top to help stimulate spawning. The eggs fall through the mesh into the trays where they can be easily collected and protected from being eaten by the adult fish. When the scientists take the spawning tray out, you can see the transparent eggs.
At Eawag, scientists use the embryo test as a basic part of research to determine the toxicity of chemicals in water. It is an aspect of the research to determine at what level chemicals are harmful to animals, the environment, and potentially to humans.
The scientists bring the eggs to their lab and place them in Petri dishes containing the test chemical in 5 different concentrations and in the control dishes prepared for the tests. In this way, the eggs are exposed from the onset before they are transferred to microwell plates, the final test vessel. Using a microscope, scientists can select the fertilized eggs and expose them to the respective concentration of the chemical. The plates are placed in an incubator during the testing period. As you can see from this film of the normal development of a zebrafish embryo, it is a fast-developing organism. The embryos are beautifully transparent, which allows you to closely follow their development. Normally, an embryo is characterized by muscles forming after 18 hours, the eye developing at the end of 24 hours, and fins after 48 hours. Every 24 hours and up to 96 hours, the test plates are observed under the microscope and the scientists record the condition of the embryos. They observe the wells to check how the chemical affects the embryo's development and look closely at 4 conditions: the coagulation of the embryos, the lack of somite formation, non-detachment of the tail, and the lack of heartbeat. The scientists call these conditions lethal endpoints. Beside the 4 lethal endpoints, they also observe and record any obvious sublethal conditions, such as heart or yolk sac edema, deformation of axis, or of the head or tail. At the end of the 96 hours, the number of dead embryos in each concentration is counted for the different time points and expressed as percent mortality. In the OECD guideline, the 96 hours time point is used to calculate the LC50. This is the chemical concentration at which 50% of the test embryos are dead. It can be calculated by using the mortality data of the Embryo Test and software programs. In toxicology, the LC50 is an important benchmark, and when scientists know the LC50 point of different organisms, they can assess how toxic a chemical is.
Our research at Eawag has helped us to show that adult fish do not have to be used anymore to test chemicals for their acute toxicity to fish. Embryos can be used for these tests, and we explore how they can further help us to predict the risk of chemicals to humans and the environment. For example, we study mechanisms of uptake and elimination of chemicals and how chemicals influence the embryo's physiology, molecular pathways and behavior. The idea is to learn what embryos can teach us about early and long-term effects of chemicals.